So today we're going to talk about DNA structure and DNA replication. First, let's talk about the function of DNA. So DNA is capable of storing, copying, and transmitting the genetic information in a cell. Super important, amazing little molecule that does this. And DNA is divided into functional units called genes. So right here would be a gene. So if all of this DNA is wound up inside of a chromosome, just one little piece is a gene. And this codes for a protein. In this example, this is a protein for making green eye color. And that protein can be expressed as an organism's traits. So you end up with green eyes. Right, so all of these features and characteristics that make up living things and how they look and how they function are all based on the sequences within these genes, within this DNA. It's pretty amazing. Now I know we've learned about DNA in the past. You definitely have learned about the structure in middle school, and we've talked a little bit about earlier this year when we talked about macromolecules. So let's give a little bit of a refresher. So DNA is a nucleic acid made of many monomers called nucleotides. So in this box here we have a diagram of a nucleotide, and it's made of a phosphate, which is this P, a deoxyribose sugar, and a nitrogen base, which is here in the center. And then zoomed in, you can kind of see them a little bit better here. There's 4 different nitrogen bases, adenine, thymine, cytosine, and guanine. So these are arranged in a different order, depending on the organism and the organism's characteristics. Each base on one strand pairs only with one base on the opposite strand. This is called complementary base pairing. So in the center of the DNA ladder, right, if you think of DNA like a ladder, those steps of the ladder are where the nitrogen bases connect. So what this means is each base, so the A's, T's, C's, and G's, only pair with exactly one base on the opposite, right? So a G is only going to ever pair with a C, and A is only going to pair with a T. This is called complementary base pairing. Okay, so like I said, adenine always pairs with thymine, guanine always pairs with cytosine. This is because of the amount of bonds that they form between them. So A and T share two bonds, or a double bond, and C and G share 3 bonds. So that's what keeps them pairing with each other. One way that I always remembered this from when I was in high school, my science teacher taught me apples in the tree, cars in the garage. Right, so A pairs with T, C pairs with G. Now let's talk about those important chemical bonds that hold the DNA molecule together. So nucleotides, remember this is one nucleotide here. They're held together by strong covalent bonds between the phosphate of one, so the P over here, and the deoxyribose sugar of another. Okay, so all the way up the sides of the ladder of the DNA, if we're thinking of it as a ladder, the sides of the ladder, so here and here, are held together by really strong covalent bonds. So these can't be broken easily. They stay together pretty well. The steps of the ladder are held together by weak hydrogen bonds, which form between the nitrogenous bases. So the A's, T's, C's, and G's. And those are represented by dotted lines here to show that they're weak bonds. And this is actually really important because, which we're going to learn about next, when we talk about DNA replication, these bases need to be easily separated in order to pull these strands apart. So these bonds can be broken and then can reform again pretty easily. Okay, so that's why these bonds are really important. They're easy to break and reform. So let's take a second to practice. So use these practice questions right now to practice the structure of the DNA. So use your notes to answer these questions. All right, now let's talk about DNA replication. So replication just means to copy. All right, so in order for cells to reproduce, they must first copy their DNA so that each new cell has a full copy of DNA. So let's think about what that means for a second. So this is a simple diagram showing one cell reproducing to make two cells. And we'll talk about this process in a few weeks. But before we learn about it, we need to learn how DNA replicates. Because before a DNA can separate, it needs to be able to replicate or copy its DNA. So if this original cell, this mother cell, has one blue chromosome and one green chromosome, before it divides, what it does is it replicates its DNA. So it makes a full copy of the blue chromosome and a full copy of the green chromosome. So now these chromosomes look like X's because they are replicated. So each half of the X is an exact copy of the other half of the X. So then this cell can grow and split into two, and each copy of the blue and green chromosomes are going to split up so that each of the new daughter cells has one blue and one green chromosome. So now these two cells that were just formed have the exact same DNA as each other and as the original cell. So this is really important when a cell is copying itself, that it can do this correctly. And I'm going to show you how that happens in a second. So here are the steps of DNA replication. First, the helicase enzyme, which is shown here, separates the DNA strands at the replication fork. If you think of the replication fork as like a fork in the road, right? So it's where the two strands are separating. So this enzyme here is just separating these two DNA strands. So it went from looking like this to looking like this. It essentially is like it's a zipper, right? It's unzipping these two strands of DNA. So now each strand of DNA, when I say strand, I mean each like half of the DNA molecule, acts as a template for a complementary strand to be made based on the rules of base pairing. So essentially, since we know A only pairs with T, C only pairs with G, if we only have one strand of the DNA molecule, we can still know exactly what should be on the other strand. So if we have an A here, what base do you think is going to attach to it? A T, right? If we have a C here, what base do you think it's going to attach? A G, right? So it's going to use those rules of base pairing to make a completely new DNA strand that attaches to this original strand. So that's essentially what happens here in step 3. So the DNA polymerase enzyme attaches free nucleotides to each template strand according to the rules of base pairing. And let's just look at this word for a second. Polymerase. Ace means enzyme. Polymer means many units put together, right? Many monomers put together. And DNA. So just by looking at the name of this enzyme, we can use these word parts to tell us that this is an enzyme that puts together polymers of DNA. So these ovals here are representing this polymerase enzyme. And you can see what it's doing. It's making this new strand of DNA based on the rules of base pairing. So it's attaching these free nucleotides that are floating around the nucleus, and it's attaching them. Where there's a G, it makes a C. Where there's an A, it's attaching a T. Where there's a C, it's attaching a G. So it's going to attach all of these until we have a full DNA molecule on this side and another one on this side. We'll end up with 2. So the results of DNA replication are that each new DNA molecule has one original strand and one new strand. So we end up with 2 DNA molecules. And these are exact copies of one another. And we can see this image a little bit better just because it's color. So this blue is representing the original strand, and the red is representing the new strand that's being made by the polymerase enzyme. Okay, so eventually as the helicase enzyme unzips this entire gene, the red molecule is going to continue to grow, and we're going to end up with 2 full DNA molecules, exact replicas of one another. Each has an original DNA strand and a new DNA strand. Okay, hope that was helpful, and please post any questions you have in the question category. Have a wonderful day.